of the big challenges in dementia diagnosis and research is the fact that there is a blood-brain barrier, so that what is happening in the brain is not always reflected outside the brain. However, the easiest fluid in which we can see what's happening anywhere in the body is the blood. It is much harder, it's more expensive to do molecular imaging with modalities like amyloid, tau, and other types of PET imaging. It is a little invasive, although doable, to take a sample of the spinal fluid, which bathes the brain and spinal cord, to see what protein or other biomarker alterations there may be. Fortunately, we are increasingly recognizing that Alzheimer's is really a systemic disease, that injury to the vessels contributes to the dementia, and that there are certain markers that freely cross the blood-brain barrier, particularly in disease when the blood-brain barrier is affected anyway. Finally, there are little pieces of cell membranes called exosomes that can break off. By looking at what proteins are on these membranes, we can say where these exosomes came from. And neuronal exosomes circulating in the blood can be a source of, again, understanding what are the biochemical molecular changes happening in the brain. Over the years, we have realized that there are specific molecular changes that are fairly typical of brain injury, neurodegenerative, and vascular. For instance, there is BDNF, or brain-derived neurotrophic factor, that increases when one does exercise and decreases when there is a high caloric diet, increases again with caloric restriction. So it may be one of the biomarkers that reflects the biological pathway that mediates the effect of a lifestyle intervention such as exercise or a change in diet with an impact on cognition. So there is other markers include things like neurofilament light or NFL that has a lot of interest recently. Markers that are specific growth factors, BDNF is one of them, but also vascular endothelial growth factor or VEGF, nerve growth factor or NGF. There are markers of specific microglial inflammation like YKL-40. There are markers of astrocytic injury like GFAP. And of course, there are the classic markers of amyloid and tau. It used to be thought that levels of amyloid and tau in the blood did not reflect what was happening in the brain. But one reason we thought that was because these are difficult proteins to measure in the blood. Their concentrations are small. Amyloid, for instance, is sticky. And so it was more assay properties that were introducing noise. Over the last decade, as we have had better assays that are able to detect smaller quantities, we have found that plasma circulating beta amyloid, for instance, does correlate with risk of future dementia. Just this year, we had a large study on over 2,500 people where blood levels of plasma tau, total tau, were strongly associated with future risk of clinical dementia as well as of subclinical brain injury as reflected in the brain size, the hippocampal volume, on performance on a range of cognitive tests from memory to executive function. So there is a lot of interest and hope that over the next 5 years, we are going to identify a parsimonious set of blood markers that could help us diagnose more accurately, track treatment modalities and their impact on the brain, and divide people into clusters of pathology or biology that might respond to different types of treatments. In the past decade, we have been able to use genome-wide genotyping to take an agnostic look at what may be the biology underlying Alzheimer's and related disorders. So we have identified genetic pathways that we had not previously suspected and are now exploring to understand fully the biology of these genes and genetic pathways. For instance, in 2010, in the Journal of American Medical Association, or JAMA, we published a paper that brought together data from the CHARGE Consortium, or Cohorts for Heart and Aging Research in Genomic Epidemiology, EADI, or European Alzheimer Disease Initiative, and GRAD, or Gene Environment Risk in Alzheimer's Disease, which was a sort of US-UK consortium. And one of the genes we identified was a gene called BIN1, which is the second most important factor for sporadic or late onset Alzheimer's disease, as far as we can tell, both in terms of the attributable risk, the size of the effect, the fact that it has an impact in persons of European ancestry, as well as persons of African American and Hispanic ancestry. This is a very interesting gene. It seems to have multiple ways in which it might be affecting cognition. For instance, it has a role in the tau pathway. It has a role in endocytic clearance of amyloid. It affects the vasculature. We recognize that certain mutations in BIN1 can actually result in a cardiomyopathy. So this is a gene that we are studying in some detail to understand what is clearly an important aspect of biology that we did not know before these genetic studies. In the same paper, another gene that we looked at was one on chromosome 19 called XOC3L2. And this is a gene that is close to the ApoE gene. So initially, it was a little challenging to see if the effect we were seeing was only because of ApoE. But we did conditional analysis and did not believe that to be so. And increasingly, there is evidence emerging that this is actually a gene that may play a role in the link between vascular and neurodegenerative processes. Jason Hinman and colleagues have looked at genes in this area. XOC3L2 is one of the genes in this area. Another one is something called MARC4. And we are not completely sure if there's only one or more than one gene in this area. But this is something that's, again, pointing us to new biology in the space between vascular and neurodegenerative processes. And I'm looking forward to seeing what we learn in the next few years.